Another rock outcrop, again, just loaded with biology.
It's a quick scene here.
Sponges,
Okay.
black corals, octocorals. Holothurians, I see a sea cucumber down to the lower left. We're moving up. This is great. Crinoids. Another beautifully flared vase sponge, just like the one we imaged a few minutes ago. Those black stalks, they're stumps that you're seeing there, are what remains of probably a dead sponge, but we're seeing on this one that substrate has been taken over by, ooh boy, trying to figure out what that might be. Looks like it might be an octocoral, and it looks like it has a brittle star wrapped around it. It's kind of hard for me to tell because the polyps are, the tentacles are not extended. And to the lower left, sorry, to the lower right, we're seeing a cup coral on the lower left. Ooh, yeah, okay, he's got to move off. We might have been a carnivorous sponge, but not sure. Okay, we are moving along. Yeah, you know what, Scott? I thought it was a bubblegum coral, too. So Scott says young bubblegum coral or an old one with missing branches. Bubblegum corals belong to a family called Paragorgiidae. Not sure. We're being asked if this is something new. This long-stalked, is it an octocoral? Let's take a look at that. Ooh, let's look at that. Scott, here we go. I'm thinking bamboo here. I see seeing a little node in there. Scott Franson from University of Louisiana and Les Watling from University of Hawaii are working on a revision of these bamboo corals. And
hi, sorry, I was in the other room.
That's okay.
So on approach, and as we're going around here, you can really see that initially this branch is like a trident. You got to see a really nice view of that. And then it puts these extra branches on at the edge and ends up kind of like a candelabra. So as you were saying, this is a bamboo coral, lyrate. We believe it's all one genus and as yet undescribed one, but we are working on it. And so it's nice to see these examples of it and these beautiful close-up shots. We'll be able to go back and look to see if the pattern of sclerites, the pattern of the polyps agrees with the ones that we have in hand. And so can say something about the global distribution. Thanks for that close-up.
You're welcome. So global distribution of the genus. Oh, looks like there's a is that a polychaete there? Sure does look like it, yeah.
Copy that, Nav.
Sorry, I'm not seeing where you're looking.
Yeah, you'll see it. You see it now. Tiny.
Polychaete or a flatworm, maybe?
Maybe. Looking segmented to me, but not sure.
Okay.
Okay.
Yeah, let's get the.
So, Scott, this is a single genus, and this new genus. So you have you're only describing one. What is the likelihood that you're getting different species within that genus, and what's the likelihood that the same?
So far, we have only evidence for one species, and if that's the case, it's quite fascinating because it means that same species is present in the North Atlantic, the North Pacific around Hawaii, and now out here in the Mariana Trench. So those are really good observations.
So now that I'm seeing so many of these Walteria sponges and lots of these stumps of the stalks of dead sponges, I'm starting to think that maybe they could be those Walteria sponges. Not sure. Very interesting.
I think we've
Yeah, go ahead,
Sorry,
Scott.
Shirley, I think
No,
we
go
may have
ahead.
seen a couple of new things. That coiled whip,
Yeah.
and we may
Can we
have
go back
just
and look at
passed
that?
a corallium or a coralliid of some kind. No need to go back. I'll just make the note. It was a fan-shaped colony.
Yeah, probably just be quick. Still at 9-0.
Hey, come on in. Yeah, go ahead, roll. Don't have a lot of time here. Well, the ship is stopped, so I have as much time as we want, but if we want to keep moving, probably shouldn't spend too much time.
Scott, where was the corallium? To the left or right of this whip?
It was before this whip. The whip we're looking at is definitely a bamboo coral that I think is different from the other one that we saw in this dive. So that would make this, I think, possibly the third bamboo coral of the dive. Bamboo coral species, I mean. But the other thing was a fan. It was missing the middle of the fan, so it kind of almost looked like two lobes. And it was before we got to this to the left. So we may be too far past it.
Okay.
Thanks.
It's over
There it is.
this guy.
Yeah.
Okay, go ahead.
Is that the one, Scott? No. No, I don't think so.
That may be, Shirley, that may be what I saw.
Yeah, maybe the brittle stars with it, made it look like it was a corallium. I don't know.
Yeah.
Okay.
This is a dead bubblegum coral completely overgrown by zoanthids.
Oh,
I don't
okay.
see any more paragorgia there. It's all zoanthid.
Okay. Thank you, pilot. We can move on.
Welcome, moving up.
Yeah, pilot, if you look at Sirius Cam, it's a little bit of a drop off.
Yeah, it's dropping off to
Starboard.
southwest now.
Yeah. Do you want your starboard swing on
We're
that?
good for right
Or, I'm sorry, port.
Covered.
Okay.
Everything is still covered with manganese oxide, but there are more patches of where it looks as if large boulders may have broken off and leaving more angular surfaces exposed.
Okay, pilot, ship has been stopped for 4 minutes.
Copy that. Nav, let's just wait till we get a little bit ahead.
For sure.
All right, pilot, you're at 7.5. How do you
Okay.
feel about another move? Or do you want to get ahead
Yeah,
a
we
little
can
more?
start.
Wow, very steep
Copy.
here.
Nav, do you copy?
Copy that.
Wow.
Same again.
Yeah, I think that'd be great.
Hopefully 2 more moves and we're on the waypoint.
Perfect.
Bridge, this is Nav.
Let's do partial.
Hello, bridge. Same again, please. 30 meters. 120 bearing and a speed of 0.2
Oh,
knots.
lots of stalks covered with manganese in among the boulders covered with manganese. Everything but this dead coral and sponge in front of us with bryozoans and no hydrozoans and brittle stars on it. Okay, biologist, Shirley's off to lunch, as you saw in the chat. So, educate me. What are we looking at? I see a crinoid and somebody sticking his arms out behind what looks to me like a dead sponge. Is this guy dead?
As the doorbell.
Okay. I thought it was a dormouse.
Wait, is it a doorbell or a doorknob? Was he dead as a doorknob?
I think
it was a doorbell. No, maybe doorknob, right?
Yeah. It's quiet as a dormouse and dead as a doorknob. But it is being taken advantage of.
Partial.
Is the lower armed creature a brittle star or a sea star? The lasers were right on the center of it.
Watch your butt.
The red ones are sea stars. The yellow one in the middle is a crinoid.
Right, and the white one below, I think, is a brittle star. It didn't look like it had tube feet.
Yeah, just careful as you back
Thank
up.
you all.
Thank you, Sean.
It's interesting to me that based on the bathymetry that we see in the quad screen, the position of the ROV, and the fact that we have a way to go yet to get to waypoint 3, which appears to be right on the edge of the really steep slope dipping down to the southeast, we are still seeing a quite steep slope in front of us. Oh, this looks like the sponge we collected coming up on the right. Is that correct?
Yep, that's the guy.
See if there's anything inside. Do a quick scene.
I'm going to take a quick snap to see if there's anything inside. And looks empty.
Who's this guy floating by? Oh, he's right in my face. Yeah, let's let him go.
We're trying to take a look at that floater, but went off too quickly. So we're seeing slightly rougher surface in some areas on the seafloor, and this may be small sized rubble pebbles fallen down slope that are only partially covered with manganese oxide. So coming into view at the top of the screen, you can see some of the smaller debris. So the manganese incrustation on the bottom of the screen is really, really thick. but where you see a bit more angular surfaces, probably that's more recent talus or broken up bits of rock that have fallen down the slope. And just a forest of critters in the background.
All right, you're driving off to my port, pilot.
Let's do a partial here.
Okay, pilots, ship has stopped.
Copy that, nav.
Very large corals in the background of that previous scene.
Yeah, let's get the squat lobster.
Rotate slightly port to keep you in view, pilot.
And we have a little squat lobster moving across the seafloor with one extremely large pincher. I'm wondering if it's lost the other one. I'll bet Scott can tell us. Okay.
Seems to only have one antenna
Diva
also.
Aman says this is missing a claw. Yep. Okay. So Diva, is this a Munidopsis? She says actually no. Take that back. So what is this guy?
Just gonna get my toe in here, Roland.
Yeah, I noticed you were sneaking in.
Yeah.
I was
Alright.
following you.
Yeah.
Tina Molodtsova tells us, nope, not a Munidopsis.
Is he eating something?
He's cleaning the hairs off his arms.
It looks like it's grooming its pincher. Or taking something the pincher has just picked up and eating it. How cool.
All right, pilot, are you good to put that next move in?
Well, we had a question
I did
from
about
Shore
45.
regarding the
How's the sonar
manganese oxide
That's me.
rate
Not
of
much.
formation
Yeah, that's you right there.
on the seafloor. And I know that Chris Kelly answers
also
that
pointed at
question very well. Chris,
9.5
are you
to
on the line?
deal.
Can you tell folks
Let me center up on you.
roughly how
All right, I'm gonna
fast
rotate
the
back to
manganese
120.
coating forms on these surfaces?
Well, there was a number that came out of an estimate off of the Hawaiian Islands. This is by, I believe, Dr. David Clegg from MBARI, the Monterey Bay Aquarium Research Institute, who did a lot of work on seamounts in Hawaii.
All right, so
And
this is
he
the point at
estimated
120.
that the manganese crust
Yeah, not much
over
in sonar.
there grew at a rate
I think
of
this last one put us at
2.5 millimeters
0.3,
on
right?
average per million years. But we also know now, I mean, I think that was back in the 1970s or early 1980s. We now know now that you can get all
You wanna
types
put
of
that in?
different
That'd
growth
be great.
rates depending on how much sedimentation there is, how current swept it is. We don't know that at least some places it's bacterially mediated. That is, bacteria are promoting the crust formations as well. So it's a very, very complicated process. But the bottom line here is that it is very, very slow, takes a long time to accumulate, and when you see a really thick coating like we're seeing now, that's an indication that this is a very old seamount.
And we are now looking at a holothurian,
Just come in tight real quick.
translucent, and you can see its gut inside full of sediment. So it's gobbling up sediment on the surface of the rocks here and swallowing that, extracting whatever nutrients it can. Yeah, we have seen this guy. Little deeper down on the slope, at least a couple of them.
Let's get an overview, I guess.
Pilot watch lead.
Yes.
I wonder if we could just take a look at that thick manganese crust that's just below the dead stalks on the right. Just a quick zoom. So we're gonna zoom in a little bit on the top of this rounded boulder that has the manganese crust, because just where the lasers are, you can see probably a fresher surface that doesn't have quite as thick an accumulation. So you can get a sense
my port swing arm, Sean?
Absolutely.
of how
And make
thick
sure you
this
have a
is. The
camera on it
surface
so you can hit
looks
the ceiling.
what we call botryoidal.
Yeah.
The term refers to
So you want to
the
rotate
surface
it
shape
inward, correct?
that looks kind of
Copy
like
that.
cauliflower or broccoli. So this is a very thick crust.
Rotating in.
That's
Do you have a
great,
camera
pilot.
on it?
Thank
Yeah,
you.
I'm using rail cam.
Yeah, but you can't see the shilling.
Oh, you want me to use pilot cam?
You can see the thick,
Zoom out.
smooth upper surface of the rock in the field of view now, just below the holothurian at the upper part of the screen. And that is very likely all manganese incrustation.
Is that fine?
That's good.
It
Come
could
back
be
in.
as much as 10 centimeters
Come
thick.
back in, Roland.
And we've been wondering what these little sort of hairy biological fuzz, as one of our shore-based scientists has named it, these little, oh, there is a carnivorous sponge. Oh, Shirley, you will be so excited to see this when you get back. So we've seen a number of these on the rock surfaces. They're very tall, very small, but they actually capture and absorb, surround with their tissues, and digest prey that they manage to catch with the arms that are sticking out from the central medallion-shaped surface of the
You're
sponge.
good, Roland.
Thank you very much, pilot. Okay, well, we can go on.
Hey, we can bring
Unless
that
there's anything on
swing arm
shore
back
anybody
out.
wants to take a closer look at. We're seeing a forest of dead sponge stumps.
Thank you, Sean.
The black
No
projections
problem.
from the surface of the seafloor are covered with manganese, but represent the stalks of sponges that have lived here in this area before.
Quick zoom.
We are zooming in quickly on, okay, is this a bubblegum coral, folks on shore? It's pink-ish. Scott? Chris? What are we looking at?
Yeah, I think it's a bubblegum coral. It could also be corallium, but the polyps look too big, so I think bubblegum.
When the polyps have closed their tentacles in, they have a much brighter color usually than what I'm seeing on this particular specimen.
Yep. Hi, Patty. It's Scott. This is a tough one, and like Chris was just kind of hedging his bets there, I'm going to suggest that this is actually a Coralliidae. Something about the skeleton looks to me much tougher, much harder, much more brittle than a bubblegum coral, than a Paragorgia coral. They're actually genetically quite closely related, so
let's get the,
sometimes it's a bit
full
tough to tell them apart. But one thing for certain that I don't see here are swellings that the polyps arise from. So that's why I'm
Right.
going to lean towards Coralliidae, but really hard to
9.75.
say.
Thank you, Scott.
Yeah, so I'm gonna jump on Scott's train there too and go with Coralliidae as well. After looking at it some more while he was talking, I would agree.
Well, thank you both. Okay.
Of course, you should acknowledge the dissenting vote. Tina Molotsovo thinks it's a Paragorgia, so it's a split decision.
Okay. Oh, I see that in the chat. Yep. We have a vote from Masako Matsumoto, who says it looks like a Corallidae.
Okay, pilots, ship has stopped.
Copy that, Nav. So how far
Wow, we
away
have
is
lots
D2
and lots
from
of
0.3?
stalks scattered over the seafloor, and I'm very interested in this sort of pebbly surface. Ooh, I see another holotherian on a rock in the middle of the screen at the top. Similar to some we've seen before. But this pebbly surface looks like individual manganese nodules forming on patches of sediment.
25 meters.
Copy.
The stern of the ship, 10 meters.
These are the kinds of things that you see on the open ocean floor as round structures composed of manganese oxide and all of the metals and other elements that are typical to be found in manganese oxide. And those, of course, are much easier to mine by simply vacuuming up the nodules from the seafloor, whereas here on the slopes of one of these giant undersea volcanoes, the manganese crust would have to be mined by trawling or scraping the material off with some sort of dredge to break it free from the surface on which it's encrusting. And coming with it would be all of the animals that we're seeing today.
Let's put an overhang here.
Starfish on the left.
Well, we have a ledge projecting out from the seafloor in front of us of rock covered with the manganese oxide incrustation. So
They're coming on starfish.
looks like it's about 1 meter tall. We have a sea star. Wow, looks like it's resting one arm on the stalk of something, and its body is very fat. And I have been told by our Smithsonian starfish expert that that suggests that its tummy is full and it's digesting. It also has a little worm crawling up the surface of it, kind of greenish yellow.
So the top of the stalk is actually rooted in. It's actually at the top of the stalk.
Yeah.
Yep.
The base is up top.
Okay, I've just been notified by our eagle-eyed videographer that it looks like that actually the
So if you
top
look under the
of
bottom right foot, there's still
this
some,
stalk may be
polyps
under the sea star, and what we're looking at right now is the attachment for the stalk. So does anybody on shore know what this
Did you
thing
copy that?
eats?
I didn't copy that.
The bottom of the sea star has uneaten polyps.
Well, the sea star is probably eating a coral, but it's a little hard to tell which one he's eating right now.
Right.
Right there. See it. Come over on top of it a little bit.
Well, we have seen an anemone eating a coral, and now we're seeing a starfish eating a coral, and I am at a loss to figure out how the starfish got hold of a coral on a stalk that long that must have been up in the water column. Unless it just wafted it down there and it was a lucky grab. What do you folks think?
So we can sort of see some live polyps right now that are extending beyond that.
Yep.
one arm there.
Lower
And
right.
it looks to me like it could be a bamboo coral, but I'm not really sure. Anyway, he probably climbed onto it and ate his way up, and he's just about finished with it. He's near the top, and he weighs so much more than the coral could actually support that the coral bent over,
I think I can
and he's about to polish
Yeah.
off the last few polyps there.
far away is the waypoint.
Oh,
I think
that's
only
amazing.
10
Looks
meters
like I'm
there,
not
so-
the only one who had a big lunch.
For sure.
Just under.
Yep.
10.
Just under 10.
Oh, yeah.
Wow.
Hey, that whole coral stalk, or almost all of it, it's almost finished. It's like going to be like a giant snake, sit there and digest for a year and a half.
That last close-up, that looked like the Candidella whip that we saw earlier, and I noted that in the Primnoidae, the family that coral belongs to, each of the polyps has little scale-like armor, or an armor of scale-like sclerites, and so apparently that armor did not do it very well against that sea star.
Not at all.
Oh, look at that.
Or maybe it is. Coming out a little bit.
Are they all arising from a single?
Come on out.
See,
All the
are
way.
they all arising from a single? They are. They're all arising from a single stalk.
Yep.
Just set up without pressure
Huh.
anything.
No, it's
Is
branched.
it?
It's,
Yeah, I know. I'm trying to figure that out
Nice,
here.
come on in.
it's,
Is that
it's-
one where maybe one or two settled on an exist? All right, Chris. What do we have going here? I've never seen that happen before.
Right, so this is a Caulophacus, and at first Henry thought that another Caulophacus had colonized the stem of one that was already there, and that was his explanation for this. But the more of these that started showing up, the more we realized that this is how this particular species of Caulophacus grows. It actually branches
it does. Look.
its stalk, which is probably quite a trick for a sponge. But in any case, we are going with Caulophacus new subgenus because we're convinced this is normal for it. This is a different species than any of the other Caulophacus that have been described.
And are they all hydroids?
Oh, there are
along this?
3 sponges on top.
Right, exactly. Yeah, 3 branches coming up.
Pulling out for a full
Oh,
sponge.
it looks almost like a 4th one might be starting.
Yeah,
wow,
it
that's
does.
very unusual.
Pulling out for full species.
And a little, is that a squat lobster sitting there?
I'm not seeing the
Looks like
lasers.
it might be.
They're on. They're just.
Okay, I see them
they're in the back.
reflecting now.
Yeah, this is pretty cool, Chris, because it looks like maybe even a 4th branch might be coming up eventually.
Sure looks like something's growing.
Yeah, how that works defeats me. It's not like a coral branching, right?
No,
I mean,
no.
Okay, video
it's a sponge
swapping out.
stock, so whatever. It's figuring out some way to do this.
And then in the lower part of the screen, is that a bubblegum coral that we're
It
looking at?
is.
Isn't that
It
pretty?
is.
I love those.
Yeah, we just saw another one earlier while you were out to lunch. And we are having a shift change of our pilot, co-pilot, navigator, and videographer. It is noon on the ship.
It is. We are coming to you live from the Okeanos Explorer, the NOAA ship Okeanos Explorer. America's ship for ocean exploration. We are exploring a place called the VOGT, V-O-G-T, GEO, in the Marianas Trench National Marine Monument. This expedition is possible as a result of many partnerships. The NOAA's Office of Ocean Exploration and Research designed and supporting this expedition, which is part of a 3-year campaign to address the Pacific Monument science, technology, and ocean needs. It's called Capstone. So we're gathering information on the abundance and diversity of living resources, as well as the occurrence of non-living resources, some of which are of commercial interest. So we need to be able to document what's there before we can manage those resources, and that's what this mission has been designed to address.
All right, watch lead, we're just getting settled in. If we could just have a minute, and so the nav, co-pilot, and I are all on the same page. I'll be with you in a minute.
Roger that. Thank you.
All right. So ship is stopped. Things are settled out.
Other NOAA partners
where are
involved
you
in this
facing?
are National Marine Fisheries, the Marine
so I
National
need to push
Monuments
out.
Program,
Okay. Right.
the Deep Sea Coral Research and Technology
So-
Program, the National
What's the
Center for
general
Environmental
bearing?
Information. The ship is operated by NOAA's Office of Marine and
you
Aviation
did.
Operations, and in just a couple minutes,
060.
we'll have our ROV crew and the videographers introduce themselves. They are from another partner, the Global
Yeah, so
Foundation
I'm over.
for Ocean
Yeah.
Exploration.
Let me come around a little bit too.
Other important partners are the many scientists, resource managers who are participating in this expedition through telepresence.
Okay. So we do actually want to.
We have, gosh, it's about 15
That's fine.
folks participating today in the chat room, probably others who are calling in from time to time, as well as many
Interesting. So we
others
started
who
kind of in the middle
are
of this
probably
feature.
probably have this running in
And then they've been moving
the
to
background
edge,
on their
so we're just
computers
getting
at
toward the edge
work.
now. So that's what we're doing right now.
No doubt. And we have about 35,000 viewers
I see
per
what's
day.
going on.
Not
Okay.
all at the same time, of course, but we
So
welcome
this is
you
kind
all,
of where the slope
especially Mike
picks up.
Basala's Gene Folk. Hello there. We're having a great time
so
out
maybe
here,
we want to line up
and
this way, then. Maybe
bringing
we're,
you live
at the
video
correct heading.
of about 100 million year old seamount.
Yeah.
Very cool.
We got 2 samples out of 4.
So the way we
Okay,
scientists,
so why don't you turn your head down and
our shore-based scientists communicate
So,
with each other is
you were saying back
over
to what they had?
basically
They
a
had like
teleconference
which I
line, a
figured.
conference
So
call
I just saw
line,
the dive
and
in
so
target,
you at
and,
home
if
can
you look
hear
at the
what it is that
ship
we're
track,
saying
you'll see
to
they're
each
making their
other.
way toward the
Oh,
edge.
Most
yeah,
of the
look at
time.
that.
So they want to
Okay.
And we
follow
also
that
have
up.
a
I guess
chat
this is it
room,
right here.
and we
Okay.
talk to each other in the chat room as well. And
So if
so
you turn your head to line up.
Patty and I are getting some information
With that
on
ridge,
identifications
you're
of
talking
things
about
that we're
what,
seeing.
0.55,
Especially me.
something like
Yes,
that?
I was just
am-
going to say,
I'm
I was
a
actually
geologist,
I was going
and,
to say, especially me, since a lot of
critters-
this stuff is biological, and my
So I'm
area of
looking, like,
expertise
on
is
the
on
edge of it here.
sponges. But I'm learning, little by little. I'm learning.
Yeah, Shirley and I, we have
20 meters. Let's bring it
distinct
out to 30
and
meters.
quite
Nice
different
please.
specialities, but what is wonderful for us is that the
That one,
people
sorry.
on shore
30 meters, yeah.
help us to identify not only the biological materials and
Yeah.
critters we're seeing, but also
Yeah, why don't you
comment
turn your heading
on
down?
surface texture,
And, well, you see,
rock
so
types,
we're actually-
the
sorry,
nature
so what I
of
meant
the
to s-
region that we're looking at
to
on
get
the
across is
seafloor.
that we're going to follow this edge right here. You see on my sonar how I'm
Yeah.
kind of looking at that? So
Okay.
we're not actually
Oh,
going
right,
to
right,
060
I
yet.
gotcha.
Yeah.
Okay.
So on the line and in the chat room right now, we have
So I'm looking at, like,
Andrea
120.
Quattrini from Harvey Mudd College,
Yeah.
Asako Matsumoto from the Chiba Institute of Technology in Japan, Chris Kelly from the University of
All right.
Hawaii,
Yeah, so
Debbie
that's what they
Blaney
had to
from
begin
NOAA's
with.
Office of
Okay.
Ocean
Yeah, yeah,
Exploration
that's where
and
they were.
Research in
That
Silver
was,
Spring, Deb Glickson from
Miscommunication
Harbor
there.
Branch Oceanographic Institute, Florida Atlantic University, and Deb was the geology
Yeah, why don't you
science
come
lead
down
on
a few
leg one
meters
of this
and,
expedition.
get
Diva
the ship moving.
Aman
Okay, let's
check.
from University of Hawaii, and Diva was the biology science lead on leg one. We have Les Watling from University of
All
Hawaii.
right, now let's get a...
And we
so
have
we wanna
Scott
continue to
France
this
from the
edge,
University
right?
of
And
Louisiana. We have Tara Harmer-Luke from Stockton College in
Okay.
New Jersey. Tina
All right, let's find
Molitsova
out where they want to go. If they want
from
to
the
continue
P.P. Shirshov
to find this
Institute of
cliff.
Oceanology
Where's the
in
next waypoint
Moscow.
down
It's
there?
up
Oh,
here.
I see it.
And let's
Dive
see,
end.
we
Okay,
have
got it.
had, I
But
think
we're moving this way
Bob
to get
Carney
to
has
this
been
contour
on
here.
and off as well, and he's
He
from
has?
Louisiana State University.
Well, actually, enough. Hey, quick question for you so we can get underway. Are we trying to head right up this like knife edge right here?
Yes.
All the way up to here? Yes? Copy that. Thank you.
So, well, are we happy just heading there now, or do they want to?
Yeah, they just want to head up.
Okay, so they do want to head 060 right now.
Yes.
Okay.
Yes.
Okay.
Would you like to introduce yourselves?
Sure. Yeah, so we just had a shift change in the front row, and we've got Andy Lister in the navigator's seat, and I'm Dan Rogers piloting right now, and we've got Don Liberatore as our co-pilot, Tara Smithy as video, and Annie White as the video clipper, and we're all with the Global Foundation for Ocean Exploration.
Thanks.
And yeah, excited to be here.
Thanks very much. And I'm Shirley Pomponi from Harbor Branch Oceanographic Institute, Florida Atlantic University, and with Deb Glickson, also part of the
Yeah, we're coming around.
Cooperative Institute for Ocean Exploration, Research and Technology. I'm the biology science lead on this leg of the mission, and I'm joined by
Patty Fryer. I'm the geology lead for this leg of the mission. I'm from the University of Hawaii,
continuing the same
Hawaii Institute of Geophysics and Planetology, and there I am a research professor. But I do teaching, and I love teaching. And part of the fun that we have out here at sea is sharing all the fantastic things that we are learning about the seafloor with all of you.
So ideally, take a look around 060. So.
Yeah, we'll look on the sonar and see what's.
So through telepresence, the technology that's making what you at home see and hear right now possible.
Okay, I'm looking at like a.
We're able to have lots of scientists, the ones we've just introduced, as well as many others engaged in an expedition. So a lot more than we could typically do on one of our, normal research expeditions.
Let's do 30 meters and keep a close eye on it.
One of the cool things about this particular research expedition is that because it is discovery-based, we are able to go and look at, see, and find things that we probably, Shirley and I probably wouldn't be able to get funded to do, because our normal protocol for getting funding for research is to propose a hypothesis-driven project that is generally focused on a particular process or looking at the environment of an organism. But here we're able to go to a variety of different types of
just take a
regions.
quick zoom at this fan coral,
we're going to look at a coral here for a sec. And so what we do here on this cruise is to provide the basis, the first step in the scientific method of discovery and observation.
Sorry?
Co-pilot, whenever you start to move, let me know. We'll double check the layback.
Are these both the same thing?
I don't think the Ziba is.
There are two corals here, right?
There are, Patty.
Yeah, so this is the same prim node that we've been imaging all day that we think is
That's good. We can move on.
Planar fan, and sometimes it has little different morphologies, so it can be a little deceiving, but I'm pretty sure it's the same thing.
Okay, we are moving on. Thank you, Chris. Syncrinoids. What looks like there may be zoanthids on that dead sponge stalk.
Oh, yeah.
Zoom in. Hold.
No, we have seen this
is
before.
good. Yeah, this is good. We don't really need any additional imagery on that. We've imaged this one earlier on the dive. Thank you.
All right. Let's zoom out, Tara.
Roger.
Thank you.
Pull away.
But, Patty, as we've been going along this dive track, just about every single vista has had lots of biology.
Really,
so really
quite different from the last one of these large ancient seamounts that we dove on, where we saw a lot of stalks covered with manganese. We're seeing some here, but we did not see the diversity and the abundance of animals in that particular area as we're seeing here. But sometime in the past, there was a huge sponge sitting on the seafloor around here somewhere, and that's what we're seeing on the bottom of the screen on the right. It's fallen off, fallen over, and has been covered by this black manganese oxide
Any interest in anything on screen right now, Watch Lead?
We've seen most of this, right?
Yes, I think we've seen most of this. Are we interested at all in the bubblegum coral? Or do you want to get anything closer on the bubblegum coral all the way over on the left? I think that's what that is.
Pretty sure. See, anybody say anything? Oh.
I think we've got images
Okay.
of
Okay.
that particular animal.
Okay.
Yeah, watch lead pilot.
Yeah.
We have seen pretty much all these animals. There's just
It's
a lot
really exciting.
of these.
It's
Yeah.
very exciting to be on this
Really.
dive where we've got a lot of diversity and really abundant
All right, so what's our direction?
corals,
right now?
different,
All right, let's
different species of
try to keep
corals,
your,
and in fact,
heading on
different
Yeah, I just came around a
classes
little
as well.
bit more.
So
Okay.
very, very
Yeah,
interesting.
it wasn't
Current must be
pushing you a bit.
or something.
Okay, I'm coming over.
Yep.
There we go.
So as we got closer and closer to this sharp ledge, you could clearly sort of see an increase in the density of the animals. And
Right.
as we cruise along the ledge, as we head up toward the top there, we'll again be on the lookout to see if the density is changing at all or if it's the same or being maintained. And we've already sort of found and identified here quite a diverse community, which is really exciting. And then trying to get an idea of how far it extends and how big it is and see if there's some other components that we haven't identified yet that are part of it.
Watch lead, what is all this debris here at the bottom?
So
we have seen a lot of that.
Yeah, so this debris at the bottom, we suspect are the branches of dead glass sponges, the stalks, of dead.
A lot of them here.
Yeah. There was another dive. I'm not sure if you were on it, Dan. That we saw quite a few of these as well.
Okay.
Dive before last.
But not nearly as much live material.
No, not nearly as much alive.
All right. So in terms of age, I mean, what can we say about this site as opposed to the one 2 days ago?
The one 2 days ago was on a very different geologic terrain.
I haven't
It
really
was
seen
on
anything.
the downdropped
I've been pushing forward.
side of a ridge on one of these seamounts. And it was on basically a downward stepping fault
I was
block.
just coming
and
down, so
we were speculating that perhaps the reason there was so
Snap zoom
much
video.
debris on the
You've
slope
seen this one? Okay.
was because we
Partial.
were likely looking at a place where the animals
Okay.
had possibly been killed by the fact that there was a big fault and an earthquake-related event that may have changed the environment in which they were living to one where they were
seriously,
not
definitely
as able to get enough food.
She's not coming down or anything?
And
Sure,
what's
here,
been
yeah.
kind of interesting from this, because I really couldn't tell on the dive that we were on 2 dives ago what kind of sponges may have made these
Right,
stalks. There's a
right.
lot of speculation,
here-
and
here
here
you
it's
get
kind of
more of an
giving me
idea.
giving me kind of a better idea of what it could be, especially since we've seen a lot of those
Okay.
sponges
I
that we're
think
calling
we're moving now.
Walteria.
Yeah, we are moving
kind of
now.
That's our return
very
from
tall,
Sirius. Remember how I was telling you sometimes use the
tubular
frame?
sponges with kind of a
It's that
pointy
line right
top
there.
and
Yeah.
projections out the side. For those of you who have been watching at home. So I'm starting to think that might be
Really?
the source of these stalks.
That's what I wondered, Shirley, because even 2 days ago when we were seeing these huge stalks, they did have what looked like a hollow interior, which would be consistent with the sponges you've just described.
So I'm thinking we'll do these 30-meter 0.2 hops and then hang out for a couple minutes and then get another one in. So let's try to be consistent.
Yeah, that means
That-
we got
Okay,
about
we're
5
coming
minutes
up on a couple of octocorals. Any interest in these before we pass over them?
2 minutes, then go again.
Yeah, I'm going to push out and wait for that move to catch up a bit.
Yeah.
And then get
I am moving, yeah.
Yeah. All right, watch lead. Well, I'm just going to cruise along, kind of get a wide view of everything, and I'm going to wait for your word to zoom on anything. So
That's perfect. Thank you.
just let me know.
Great. Thank you, pilot. Okay, so we're going to be
About 5 minutes.
cruising along.
Roger that.
I'm
Copy.
looking at this vista of, oh my gosh, look at all of these fan corals. Sponges, another one of those
there's another-
sponges.
Yeah,
that's
another one.
branched.
Another one like we just saw before. Yeah.
Wow.
Very, very interesting.
Folks,
Really, I've
yeah,
never
if
seen that.
anybody's
sponge.
just tuning in,
Huge
huge.
sponge in the background there. Paddle
Huh.
sponge. Yep.
for people just beginning to look at this branched sponge in front of us with 2 pom-poms on the top, people were talking about the fact that they don't really know how a sponge can branch like this. This is not typical, so I'm learning a lot.
Well, do we want to look at it, or?
No, we
We did see one that
All right.
Yeah, we
had
just looked at one.
3 poof balls on top.
Well, you know what? This one looks like it's getting another one on top of it as well.
It does.
Try to give you a nice
Very
pan.
cool.
This is a beauty.
Oh my
this,
gosh.
This is a beauty shot.
Yeah, so we don't have to zoom in to do beauty shots.
Yeah, you don't
Yes.
have to. You don't necessarily have to. Yeah, you don't necessarily have to zoom in to
could
do a
frame
beauty
it,
shot.
yeah.
This is gorgeous.
All on its
I'm
own.
going to tilt down a bit.
But
We can kill lasers. I think
Roger that.
we got an idea there. Yeah, see it? About 10 centimeters wide right at the middle there.
Very pretty sponge. And it
This
has
is what
a
I believe
crinoid
that
on it.
Chris called the paddle sponge. Paddle.
All right, I'm going to move around it here. And it's
a
pretty
golden
flat now,
crinoid.
so let's
Beautiful.
get another move in.
Really pretty. Lots of small things now I can see growing on the rocks. Smaller
No, I
branches.
mean, I think it's so flat here, I think we go with what does
If,
HiPAC say from here.
if
So the
people would
same
like to take a look
060.
at the center of the 3 feeds on the bottom of your computer screen, you will see D2, our ROV, in a view from the
Let's say it's
Sirius
70.
vehicle, which is
Another
attached
30
above
meters,
it on a
70.
30-meter cable. And in this way, we both can see
Copy.
the regional geology, regional structure or form or extent of the seafloor. We can also monitor the vehicle, just to make sure everything looks like it's working, all the lights are on, and there aren't any large sharks in the background headed for the cable or anything.
You mean mine
Did you
personally?
say sharps or sharks?
I
don't
said
know. How's
sharks.
this any better? Copy.
We have sent vehicles down to collect samples from the seafloor and seen
All right, actually,
sharks going for the floats on the umbilical cord
this might be a good opportunity
on the cable.
to have you jump in, Don.
Oh, okay, sure.
So one of the goals of this
Yeah.
particular dive was
Some height here.
to
Okay.
explore for high-density communities of deep-sea corals
not
and
even a lot of
sponges,
currents.
as well as characterizing the manganese crust habitats. This is one of the oldest seamounts on the Pacific Plate. So, indeed, we are seeing manganese-crusted rocks, and we are definitely seeing high-density communities of corals and of sponges. There's already a mining lease that's been issued by the International Seabed Authority for the nearby Magellan seamounts that are located just outside of the U.S. Exclusive Economic Zone. This is within the U.S. Exclusive Economic Zone. And so what we're trying to do here is to get a handle on the resources, both living and non-living, in our nation's monuments and within the exclusive economic zone of the United States, so that we can better manage these resources.
The Magellan
You're all set?
seamounts really
Yep.
are just east of here and are very similar to what we're looking at today.
So, watch lead, just in case you didn't notice, we just had a pilot swap. So we got Don in the pilot chair now.
Roger that.
There are many
Roger
fewer
that.
critters in this particular area. The seafloor is
Yeah,
much
this is
flatter
kind
here.
of interesting here.
And we just had a bit of a shift change. Same individuals. Don Laboratory
Yeah.
is
That's
now
all
our
the tether you
pilot.
got.
Dan is
Dan
moved
just
over to
moved laterally,
co-pilot. And Andy left. And who do we have?
No, Andy is there.
Andy's
Oh,
there.
is that
Fernando
Andy?
stepped
Oh,
out.
sorry.
Yeah.
No, Andy is still there.
Okay.
We just had a little switch.
I think maybe there was somebody else come in and confer with Andy. Interesting slabby seafloor over here, and an errant boulder.
We are at 1,807
So right now your camera's tilted up at 7
meters.
degrees, just so you know.
Okay.
Which is totally fine, especially on this slope, like you want to see in front of you, right?
Yeah.
But before you make like quick turns,
And one
just,
poor
glance
little
up at your
remnant of
manipulator
a something
camera.
Yeah, that's
here.
what, actually, that's
Yeah.
what I was looking at.
Yeah.
Looks
And then
like
your
there's
starboard
a bat.
Port rail
Is that
cams
a
can kind of give you an idea of the slope up
in
ahead.
front of us?
Okay.
Not sure.
There's a fish in Sirius.
Oh,
Still learning
yep,
them.
Yep. You mean the bottom of the screen? Yep.
Yep.
And the mid
Is that
screen.
something there you want
That
to look
is
at?
Screen. No pilot.
Nope. Okay. Roger that.
What time did you call that move in?
And if you look at
Hey,
the
this is Deb. If you guys have a
018.
chance, you might want to look at the stereo view. There's a fish wandering around in there.
we're seeing that.
There is a fish.
I can call another one in about
Right.
3 minutes
It's
here.
not a shark,
Give us
though,
some time
Deb,
for
so I think
layback
the
to get out a
cable's
little bit.
okay. But there is a
Reduce our
fish
layback.
next to the cable.
Reduce our layback.
Yeah, if I
Ah,
give
okay,
it another 3
Holothurian
minutes, I
on the
mean,
far
Sirius will have cut
left.
up a little bit more, so that layback will have decreased.
Okay,
Call another
large
like we did.
branch.
Okay.
Kind of what we discussed.
Yeah.
Pilot, can
yeah.
we go in and take a look at that large
fine.
branching coral, please?
The lower left?
Yes, lower
Okay,
left.
Roger that.
We're seeing a Holothurian on the right, or actually center right now.
And we are seeing a distinct
Moving back
change from
toward
the
me.
flat
Yeah.
surface we were on
Okay,
just
so
a
they
moment
wanna
ago
look at
to
this,
downsloping
branching
to the
coral here.
left.
Okay.
Or whatever
And
it
at
is.
the top of the break-in slope, there looks to be
So Sirius should just about
fracturing
be starting this second move.
of
No, I'm
the
definitely
seafloor.
moving.
No, I mean it's under the influence of this latest move now.
Okay.
That fish
So
is
what
moving
was the
toward
break
the
between
front
the previous
of
two
the
moves?
vehicle.
I'm going. Oh, the break between the two moves was 9 minutes.
Okay.
8 to 9 minutes.
I wonder
let's
how
not
high that
hang out
fish
here
is.
too
Looks
long.
like it's
Okay.
pretty high
Roger, you
above
want
the
to
Yeah,
vehicle.
might
zoom
be.
in on that video, please?
But it's moving.
So there were.
Ooh,
Yeah,
wow.
I just wanted to take a quick look at this.
Okay. Something on a dead.
No, I don't
Stalk?
know. I don't. You know,
Looks like
it's
parts of
part
it's
it are
alive.
dead, parts of it are.
There's something in
Yeah,
the
you don't
Oh,
have
and
much
there's
time.
a
there's
jelly.
a jelly in the
What's that
background.
You don't have much
Oh
time.
my gosh.
Yeah, okay.
Oh, we got to zoom on the jelly.
Yeah.
Right, Shirley? Don't we?
Okay, we
Don't we?
gotta watch
Like
this. We
jellies.
gotta move in already.
Can we
so
zoom on that jelly?
Oh,
All
it's cute.
Yeah, let's do a snap zoom.
Snaps in.
Okay to zoom.
Let me just rotate it.
Roger. Standing by.
Okay, go ahead.
All right, Don.
That's
Let's.
not enough. Okay, time to
Yeah.
go.
Yeah, we need to push out.
Roger.
Yeah, we need to push out.
Yeah.
We're on the move.
So lift up and
Yep.
push forward.
Almost looked like a dandelion.
It looked like
Ah,
it was
fish is
sort
gone.
of stuck on that stalk as well, huh? Okay.
Yeah. So
Oh, that
lateral
jelly was
starboard,
hanging out in a spot on the slope of the seafloor facing us that's just at the bottom of the screen, and it looks like a
Yeah, you can
kind
really
of a
punch it
hole
forward.
where part
Oh,
of the
okay.
crust
Put some juice to it.
of
Yep.
the manganese
Sure, I'm up high enough here.
has
before I
fallen
do that.
away and exposed some inner parts of the rock surface.
And yeah, you're looking up at 8 degrees. So
Yeah.
I'll show you the angle that I typically like to fly. I'll tilt your camera down a bit. It depends on the slope, obviously, but
Yeah.
just like a somewhere around
30
minus 10.
degrees or so.
So
Oh,
Diva
really?
is
Yeah.
thinking that the
If you go down to like 15,
Mhm.
you'll see just the tip of
what
your
we thought
skids,
was a
which gives you a little bit of
jellyfish
perspective. So just above
before
that is where I like to
might
fly. But
be
Something
It's a
else?
little
Let me see.
What's she saying?
Dandelion. Yes, a dandelion
Wow.
yeah, so it's
siphonophore.
really
It's
let's
stationary, so that
stretch out
means that it's
ahead a bit.
anchored, and
Gonna
that's
come up.
typical of the dandelion. So it was not a jelly.
Ah.
Diva and
this would be a
Deb saw
typical
some
time
of
I call
these
the next
on
move.
the
You wanna give it a
leg
minute?
Yeah,
one,
let's give it a minute.
and
Yeah.
it was really exciting. And I think we saw one right at
Yeah.
the beginning of our leg as well. So, all right,
And they
ladies.
are beautiful.
So right now,
oh, look at
our
this
two
fracture
we're in
going
the move
right
that you
one.
just
up
called
the
previously.
center
Yes.
left of the screen.
Got it. That's good.
And I mean, you'll have at least.
It's
3
not
to 4
surprising that you
minutes
might
left
see a
of this.
fracture
Yeah.
like this close to the edge of a very steep
What's that thing
drop-off.
on there?
Which, Andy?
Okay, we
Something
got it.
on the...
Yeah, I just saw that little
yeah.
black thing hanging out.
Yeah, it looks like it might be a brittle star on
Oh, okay.
the sponge stalk. That's great. So I'm seeing quite a few of these kind of what look like they might be dead branches or branches of these, what did you call them?
Oh, neat.
Candidella, I think it was Candidella. Okay, this is a Walteria. So this is one of these hexagonal sponges that we've been seeing quite a few of.
Time,
Yep.
That may be producing some of these stumps of stalks.
It might be what those stalks are, because I was having a
Some
hard
of them
time the
anyway.
other day figuring
All right,
out
let's get another move in.
if that was just a stalk of
It's
a
another
big
one?
sponge.
Right in
yeah,
the middle?
one of them.
Okay.
That was just a stalk of one sponge on which the sat actually most of the mass of the sponge. That would have been a very long and thick stalk.
I think
Now, having seen all these Walterias here, I really had a different idea of what it could be.
Hey, same move. 30 meters, 070, 0.2 knots. Sorry to disappoint.
Well, we have seen a lot of diversity, but we're also seeing abundance like we have not seen on one of these seamounts before that I remember.
Yeah, I agree. But you know what? We haven't seen very many fish, have we?
Well, I think we've seen 3 or 4, and then there was the one swimming around between Sirius and the ROV just now, but that's not a lot.
Many, no, that's
actually, we saw more
many.
on the really deep dive yesterday than we have seen on this one.
Previous
Which is a
moves probably settled
surprise.
out. Yeah, so just have a minute.
Yeah, it should.
But if we stay
If it's
close
not
to the
30
edge
seconds.
of the scarp,
Yeah, I
we'll
think
probably
it is
see
already.
more.
So have